Today, we'll be examining a recent paper from Aging Cell that investigates whether Memantine, a drug currently approved for neurodegenerative conditions, might also function as a caloric restriction mimetic. The researchers used the model organism elegans to explore Memantine's potential to extend lifespan and mimic the well-documented effects of dietary restriction. So, let's look at their findings. Our discussion will follow the logical progression of the study. We'll begin with the rationale for investigating Memantine. Then, we will review its effects on lifespan and reproduction, followed by an analysis of the transcriptomic data that links it to caloric restriction. After that, we'll look at its role in modulating cellular stress, and finally, connect these molecular changes to shifts in metabolism and behavior. So, let's start with the two main components of this study, the compound Memantine and the model organism C. elegans. This nematode is a principal organism for aging research due to the conservation of many of its genetic pathways with those in humans, which makes it a useful system for longevity studies. Now, Memantine itself is not a new compound. It's an established clinical drug that functions as an antagonist of the NMDA receptor. Its primary mechanism is thought to involve the mitigation of excitotoxicity in neurons. This study, however, poses a different question. Do its biological effects extend beyond neuroprotection to influence the systemic processes of aging? The first objective was to determine if Memantine exposure had any effect on key aging phenotypes. To do this, the researchers measured two standard parameters in aging research: lifespan and reproductive output. These metrics provide a direct assessment of any potential pro-longevity intervention. The results were notable. Worms treated with Memantine exhibited a significant extension of their median lifespan. What makes this particularly interesting is the comparison with ketamine, another NMDA antagonist. At the tested concentration, ketamine had no effect on lifespan, which suggests that the pro-longevity effect is specific to Memantine and not a general feature of NMDA antagonism. In addition to lifespan extension, Memantine also extended the reproductive period of the worms. Control worms typically stop laying eggs around day 4 of adulthood, but the Memantine-treated worms continue to reproduce beyond this point. What's particularly informative is that this effect persisted even in mutants lacking the NMDR subunits, NMR1 and NMR2. This strongly suggests that Memantine's impact on reproduction operates through an NMDR-independent pathway. So, we have a clear phenotype, extended lifespan and reproductive health span. This raises the question of the underlying molecular mechanism. To investigate this, the researchers employed RNA sequencing to analyze the changes in gene expression induced by the drug. The transcriptomic data further distinguished Memantine from ketamine. As this table shows, Memantine induced a much more substantial transcriptional response, with nearly double the number of upregulated genes compared to ketamine. This finding reinforces the idea that Memantine possesses a unique biological activity not shared by all drugs in its class. And here we come to the central finding of the study. The analysis revealed that the genes upregulated by Memantine showed a significant overlap with the transcriptional signature of the EAT2 mutant. The EAT2 mutant is a well-established genetic model for caloric restriction in C. elegans. Key genes in this overlapping set, such as ZIP2, UGT44, and GST22, are involved in stress response pathways, including detoxification and resistance to mitochondrial stress. This similarity strongly suggests that Memantine may be activating the same protective pathways as caloric restriction. The transcriptomic data suggest an upregulation of stress resistance genes. The next logical step, then, was to functionally validate this. Do these changes in gene expression actually translate into an increased capacity to handle cellular stress? The first pathway they examined was the mitochondrial unfolded protein response. For this, they used a reporter strain in which the promoter of the mitochondrial chaperone gene, HSP6, drives the expression of green fluorescent protein, or GFP. Increased fluorescence in this strain indicates heightened mitochondrial stress. And the results were quite clear. The Memantine-treated worms showed a significant reduction in the HSP6 GFP signal. This indicates that Memantine mitigates mitochondrial stress. This is an important finding because it provides functional validation for the insights gained from the RNA sequencing data. They extended this analysis to other stress pathways. Using similar reporter strains, they found that oxidative stress was also reduced. However, stress in the endoplasmic reticulum monitored by an HSP4 GFP reporter was unaffected.
This demonstrates that memantine does not act as a general stress suppressor. Rather, it selectively modulates specific stress response pathways.
So far, we've seen effects at the genetic and cellular levels. Now let's see how these changes manifest in the physiology and behavior of the whole organism.
Memantine also induced distinct behavioral changes. Treated worms displayed delayed food-seeking behavior after starvation and also exhibited an increased average locomotion speed. What's notable here is that the increase in locomotion was abolished in the NMR1 and NMR2 mutants. This contrasts with the reproductive effects we saw earlier, and it provides evidence for memantine's pleiotropic effects, acting through both NMDR-dependent and independent pathways.
Increased locomotion requires a greater energy supply, which naturally leads to the question of how the organism's metabolism adapts to support this higher energy expenditure. One might hypothesize that the worms would catabolize their lipid stores. However, a reporter for lipid content showed no significant difference. Instead, the researchers observed an increase in the activity of a reporter for fatty acid beta-oxidation. This suggests that the worms adapted by upregulating their capacity to metabolize fatty acids rather than by depleting their existing lipid reserves. All right, let's synthesize these findings. We've examined the effects of memantine on lifespan, gene expression, stress resistance, and physiology. So what is the concluding picture of its mechanism of action?
To summarize, memantine extends lifespan and health span. Mechanistically, it induces a transcriptional profile that significantly overlaps with that of caloric restriction. This translates functionally into increased resistance to mitochondrial and oxidative stress. And finally, these effects are supported by a physiological remodeling of metabolism toward enhanced fatty acid oxidation. It's a cohesive narrative from the organismal phenotype down to the molecular mechanism.
The authors themselves summarize it this way in their paper. They propose that memantine enhances stress resilience and promotes healthy aging through metabolic alterations that are associated with caloric restriction pathways, positioning it as a potential intervention for modulating longevity. The major implication of this research is that a compound known for its neuroprotective effects may have systemic pro-longevity effects that mimic caloric restriction, at least in a model organism. This work bridges existing data and provides a whole organism perspective, which raises an important question for future research. Could targeting NMDR-associated pathways represent a viable therapeutic strategy for longevity interventions in humans? This study provides a foundational step in that direction.